Welcome everyone to today's presentation. My name is Chowdhury Siddiqi and I will be presenting our paper titled Nuclei Instant Segmentation of Cryosection HNE stain histological images using triple unit architecture. Me along with my co-authors would like to thank the organizers of the 6th International Conference on Imaging, Vision and Pattern Recognition for giving us the opportunity to present our paper in front of our esteemed audience. So first of all, I would like to start off by briefly explaining what is nuclei segmentation. So nuclei segmentation is a process by which we segment and identify the regions containing nucleus in cell images. So these are some of the example of cell and nucleus and the tissue samples. So nuclear segmentation has a range of application distinguishing between benign and malignant lesion, online cancer grading, determining borders around abnormal tissues. So before examining a tissue sample, it is pre-processed. There are two pre-processing methods that produce frozen sections which are fast to produce but the samples are of low quality. Then there are FFPE sections which takes longer to prepare and makes samples of very high quality. So frozen sections images or the images taken of these frozen sections are of very low quality as the samples are of low quality by themselves. So the problem that we are tackling is to develop a computerized automated system that can take as input an image of a tissue sample that has been pre-processed by the frozen method and identify the nucleus in them accurately and more faster. So for background study, we have gone through the Cryosec paper which introduces the data set that we have based our research on. It contains 30 images of 10 organs that has been annotated by 2 annotators. Also we went through the UNET paper as the baseline architecture has been using the UNET model for performing this task. The UNET is a basic encoder decoder model where the input image goes through a downsampling and upsampling process to produce the segmentation maps. So there are some problems with the UNET as it is not a domain specific model. It is not that good at detecting overlapping nuclei. Also the pixel wise recognition performance is not up to the mark. Also the feature extraction or the contextual information extraction process is not suitable enough or not up to the mark for this sort of complex image datasets. So our proposed framework, we want to replace the UNET model with the triple UNET model. So in the triple UNET model, as you can see here, there are 3 branches: the RGB branch that focuses on extracting raw features, the H branch that focuses on edge detection or contour detection, and the segmentation branch that merges these extracted raw features and contour features. Also here it follows a progressive dense aggregation method for concatenating the feature, which is quite different from the baseline UNET model. Here as we can see on the left, we have the baseline architecture used in the baseline paper using which the benchmark scores have been developed, and on the left on the right we have the proposed architecture using triple UNET. So here as we can see, the image goes through the triple UNET, the segmentation results are then passed through a watershed post-processing that further enhances the segmentation mask by separating the overlapping regions. For evaluation metric, we have used the aggregated Jacquard index and the panoptic quality. These are the scores that we achieve across 10 classes for both AGI and PQ. These are the comparison score of the PQ scores. As we can see in bold here, the average score across all 10 classes is higher using our proposed framework. Also in case of AGI score, we achieve a better score which is much higher compared to the PQ score difference that we saw before. AGI specifically is quite a strict metric which actually penalizes the wrong predictions more. So performing better on the AGI score makes our proposed framework more reliable. We have further compared it to other models that have been implemented on this dataset of frozen samples, and as we can see, our proposed architecture using the triple unit model and watershed post processing performs better than those models too. So if we summarize the overall performance, our proposed architecture outperforms the baseline model and also other models that have been implemented on this frozen dataset of frozen samples across all the evaluation criteria such as AGI and PQ. Also, the proposed triple unit architecture does not require color normalization, which saves the computation power, and it is also better at edge detection. But still there are room for improvement, as the triple unit model that we used in our proposed architecture performs below par for some of the organs. This is partly due to the small dataset size and also the low quality of images that frozen sections normally produce. So in conclusion, this framework that we propose using this deep learning model called triple UNet along with watershed post processing, so it can be used in automated medical systems to segment nucleus in cell tissue images, and it can take out the manual intervention and automate the whole process. But still there is scope of improvement to improve the accuracy by increasing the data set on which the models are trained or which the framework is trained, also using newer state of the art augmentation techniques and newer models can also enhance the accuracy and performance of this framework that we have introduced. So these are some of the references that we included. Thank you everyone for giving your time, and I'm happy to take any questions from the audience. Thank you.